Welcome to Lecture 14. This lecture will go over cell communication. Cells can communicate with themselves and with each other through long and short distance signaling. In long distance signaling, a cell will send a signal that can be received by other cells in a different region of the body. This is also known as hormone or endocrine signaling. Short distance signaling is when the signal only has to travel a very short distance. So this occurs when a cell is either signaling to itself or signaling to adjacent cells. When a signal is acting on the same cell that produces the signal, that is called autocrine signaling. Paracrine signaling, on the other hand, is when a cell sends the signal and the neighboring cells in the close vicinity will respond to that signal. Within these short distance signaling pathways, you can have a local regulator. A local regulator refers to the actual signal molecule that is causing the response, either in itself or in neighboring cells. An example of a local regulator is a neurotransmitter. A neurotransmitter acts between two communicating cells in the nervous system. A ligand, on the other hand, can refer to the signal molecule, whether it's short distance or long distance signaling. So a ligand can be used to describe either a hormone or a local regulator. This diagram here goes over the different types of signaling. In paracrine signaling, which is an example of local signaling, here we have the signaling cell releasing signal molecules that have an effect on the target cells in the near vicinity of the signaling cell. Notice how these signal molecules are released from secretory vesicles. Once again, I'd ask you to recall the importance of the endomembrane system, because this is the system that would produce those signal molecules and package them in the secretory vesicles to be secreted outside of the cell. In this instance here, we have a local regulator signal molecule. In synaptic signaling, which is part B of this image, we also have a local regulator. In this instance, it would be the neurotransmitter, which diffuses across the synapse to cause a response in the adjacent target cell. In endocrine or long-distance signaling, or also known as hormonal signaling, we have the endocrine cell, which produces the hormone. It releases the hormone into the bloodstream, which then causes an effect in the target organ. So we can see in long-distance signaling, the target tissue can be very far away from the actual cell producing the signal. You can divide up your ligand molecules into two different types, depending upon their characteristic features. You can have hydrophobic and hydrophilic ligands. Hydrophobic ligands are water insoluble. These include your sex steroids, cortisol, aldosterone, and thyroid hormone. Hydrophobic ligands, in general, work very slowly. These ligands are more common in long-term regulation and homeostasis. In hydrophobic ligand pathways, receptors can be inside the cell in the cytosol, and this is because, if you remember membrane transport, hydrophobic or nonpolar molecules can easily cross through the cell membrane. As a result, the receptors for these hydrophobic ligands will be inside of the cell. Hydrophilic ligands, on the other hand, are considered to be water soluble. These include the vast majority of hormones and local regulators. Examples of these can include peptides, histamine, epinephrine. Hydrophilic ligands can work very quickly if needed. This is because they use secondary messengers to help spread the message across to the cell very quickly. Hydrophilic ligands, because they cannot cross the cell membrane, require a receptor on the plasma membrane surface. This slide gives an example of your two different types of signals. Hydrophobic ligands being water insoluble. Common examples are your steroid hormones, as we can see here. Recognize the four-ring structure that is common for steroid molecules. Here we have aldosterone as well as estrogen. In hydrophilic ligands, these are considered to be water soluble. So these would require receptors on the cell surface, examples such as epinephrine, insulin, and neuropeptide. Notice that neuropeptide and insulin are both considered to be peptide-based or protein-based signal molecules, whereas epinephrine is not protein-based. However, we can see that it has a lot of oxygen groups, which make it a polar molecule. We are now going to take a look at the pathway that these hydrophobic ligands take in the cell. When a ligand is hydrophobic, such as your steroid-based hormones, once again, the ligand can diffuse right into the cell. This is because it can pass through the hydrophobic barrier that is present in the lipid bilayer from the tails, the hydrophobic region. This does not provide a barrier to these types of signal molecules. So these ligands will pass directly into the cell. When the ligand enters the cell, it will bind to its receptor that is in the cell cytoplasm. Then it will form, upon binding, a hormone receptor complex. This hormone receptor complex will then pass into the nucleus through the nuclear membrane, specifically through the nuclear pores, and it will act as a transcription factor. This is important because it will be able to either turn genes on or off. It can also up or down regulate their transcription. So it can directly affect the transcription of genes. In these pathways, no secondary messengers are needed. We will see what secondary messengers are and their role in signaling pathways when we look at the pathway of hydrophilic ligands. There are many steps in signal transduction pathways. These involve the first phase, reception. This occurs when the signal is detected by binding to its receptor. This will then lead to the transduction phase, where the signal will cause a change in the receptor to initiate a pathway inside of the cell. This pathway usually involves a phosphorylation cascade, which we will see when we take a look at hydrophilic pathways. And finally, you have a cellular response. This is when the cell responds with a specific cellular action that is specific for the signal molecule or for the pathway. This can either be an activated enzyme, it can also be a nuclear response, such as turning on a gene. Here we have an overall example of the general pathway that is involved in a hydrophilic molecule. The hydrophilic signaling molecule, remember, is hydrophilic, so it cannot pass through the cell membrane on its own. As a result, it will bind to its receptor on the cell membrane. This initiates the reception phase. This will then cause the transduction pathway to occur through a series of relay molecules. And finally, this will then cause a cellular response. Remember, this can be an activated enzyme. It could also be some sort of nuclear response, such as turning on a gene or up or down regulating a gene. When we are dealing with the reception phase, remember that ligands will only affect cells that have the proper receptor. This means that a cell won't be able to respond unless it has and is producing the proper receptor, and that receptor is open to receiving a signal. This is one of the ways in which you can have tissue-specific responses. Most of the receptors for the reception phase are on the plasma membrane. These would be for the majority of your hydrophilic ligands. Many of these pathways, that involve a receptor on the plasma membrane, will use a secondary messenger. This will pass the message to the cell very quickly. Some receptors, however, will be inside of the cell in the cytosol. Remember the pathway for your hydrophobic ligands. Thus, these are called intracellular receptors. We're now going to talk about the different kinds of membrane-bound receptors that you can have. These types of receptors are going to be present when your ligand is water-soluble or hydrophilic. This is because, remember, they cannot pass the cell membrane. These would be common in your peptide hormones, such as insulin. There are three main types of membrane-bound receptors that we can have in these pathways. The first is G-protein linked. These are receptors that will be linked to a G-protein intermediate. The second type of membrane-bound receptor is your tyrosine kinase receptor. These receptors are also enzymes. The term kinase can be used to refer to an enzyme that typically will phosphorylate other proteins. As a result, the tyrosine kinase receptor will phosphorylate other enzymes or other proteins in the vicinity when it becomes active. The third type of receptor is your ligand-gated ion channels. These receptors are also ion channels, which means that the binding of the signal can open or close the channel to allow or prevent ions from passing through the cell membrane. This slide depicts the action of a G protein-linked receptor. Let us start with the first image here, which is to the top left of the diagram. We have our G protein coupled receptor. This can also be termed a GPCR. We have a G protein nearby, which is present in its inactive form. When a G protein is in the inactive form, it is bound to GDP. If you remember, we have ATP and ADP. So when we have GDP or GTP, this is just very similar to the structure of an ATP molecule. It can easily be hydrolyzed to form GDP, or it can easily have another phosphate group attached to it to form GTP. And also within the membrane, nearby to the receptor in G protein, we will have our target enzyme. In the second image here, which is the top right, we now have ligand binding. So our signal molecule, which must have been hydrophilic, will bind to the receptor in the cell membrane. This will cause the receptor to become activated. This will join to the G protein, which is close by, and it will activate it as well. When a G protein becomes active, it will become bound to GTP. As a result, the GDP molecule will leave, which is what we see in the diagram. And now the G protein is active and is bound to GTP. In the third portion of this image, in the bottom left, we have the activated G protein sliding through the membrane and binding to the target enzyme. This will then activate that target enzyme to cause a cellular response. In the last portion of the diagram, we have the inactivation of the G protein. As a result, the inorganic phosphate group that was on the GTP molecule will be removed and GDP will remain. In this phase, we have the return to the initial state. This image here depicts the action of a receptor tyrosine kinase. In the first part of the image, once again in the top left, we have the ligand binding to the receptor. Note that the tyrosine kinase receptor is present in two separate units. Also notice that we have many tyrosine residues that are on the receptor itself. These are important because this is where the phosphorylation will occur. When the ligand binds to the tyrosine kinase receptor, the signal molecule will come in, bind to both receptors, and form a dimer as seen in image 2. This dimer will then become activated, meaning that it will become phosphorylated, which we can see in image 3. Once these phosphorylated inorganic phosphate groups are added to the tyrosine kinase receptor, these phosphate groups will be coming from ATP molecules. So ATP molecules will be hydrolyzed. Those inorganic phosphates will then become attached to the receptor. Once we have an active receptor that is bound to all of the inorganic phosphate groups on the tyrosine residues, we can see 6 of them bonded to the receptor. These inorganic phosphate groups will then be transferred to the relay proteins. Once they become transferred to the relay proteins become active to cause a cellular response. Once they leave the receptor, they become inactivated and will no longer pass on the response. This is the action of the tyrosine kinase receptor. Finally, we will take a look at the ion channel receptors. In this case, we have a signaling molecule. Once again, this would be a hydrophilic signal molecule. it will bind to the ion channel. In this case, once the ligand binds, the channel opens to allow for the passage of the ions to the interior of the cell. These ions will then cause a cellular response. We can also see that when the ligand is removed from the receptor, the channel becomes closed once again to prevent the passage of these ions. Please keep in mind that this is only one way that these ion channels can act. You can also have them open in the natural state, and ligand binding would then cause them to close. And conversely, when the ligand would be removed in that case, the channel would then become open again.